Olá, boa tarde. Então, dando continuidade aqui às palestras da tarde de hoje, eu queria apresentar para vocês o Dr. John Moore. Ele é professor de microbiologia e imunologia da Faculdade de Medicina da Weill Cornell, na Cornell University, e o trabalho dele é voltado para a utilização do conhecimento da estrutura e função do trímero da glicoproteína do envelope do HIV para projetar e avaliar formas recombinantes dessas proteínas como imunógenas. Inclusive, o que ele vai falar para a gente hoje um pouco é o papel desses trímeros no desenvolvimento e na produção de vacinas. Welcome, Dr. Moore, please.
Thank you. Hello, and thank you all for being here, and thank you for Esper and his colleagues for arranging such a lovely meeting. We've all enjoyed ourselves tremendously. I know I have, and I really appreciate being here. So I'm going to talk to you today on a vaccine-related theme, following on from what David Watkins and Ron DeRozier talked to you about this morning. But my focus is going to be on antibodies, how to induce neutralizing antibodies, because my colleagues and I think that if we can achieve the induction of strong and broadly neutralizing antibodies, that is sufficient to make a protective vaccine. We may not succeed, but that is our goal. And I'm going to tell you about what we call SOSIP trimers, which are a platform for immunogen development. They are a protein system that we can modify to try to improve the immunogenicity of the envelope glycoprotein trimer. And as with everything nowadays, it's a team effort. And here we are coming from a lab meeting. This is what we were in America nowadays. We're very formal people. This is Ian Wilson, Rohir Sanders, Andrew Ward, and myself. And everything is on behalf of us all. We do it as a team. So what are SOSIP trimers? They're recombinant soluble protein mimics of the envelope glycoprotein trimer on the virus. So we have 3 GP120 and 3 GP41 subunits, and they're linked together by a disulfide bond that we engineered into the right place, and a point substitution in GP41 that stabilizes the pre-fusion form of the trimer. The proteins are fully cleaved. We can achieve that by co-transfecting a furin protease enzyme. So we can make them in a form that is cleaved and still stable and resembles the virus spike. They've been used to generate high-resolution structures by X-ray crystallography and cryo-electron microscopy. We can nowadays make them from most genotypes because we've accumulated enough knowledge of the stability determinants so that we can further engineer the structure to make it from a Brazilian virus, a Thai virus, a Chinese virus. We can do it essentially for most viruses. They can be made in gram amounts under GMP conditions, and they're now used as the basis of immunogen design and GMP programs by the best funded groups in the field, which is not us. The Vaccine Research Center uses this, all of the 3 CHARVID applications, and 2 EC-supported research consortia. So they have far more money than we do, but we try to keep on setting the field in the right direction, and then they just copy us. So the cleaved trimers, you can take a negative stain electron microscopy, and this is done by Andrew Ward and his group, and you can see that they are regular structures. They look like trimers. But the earlier generations of proteins that were supposed to be trimers, you can see that they're all sorts of weird shapes. They don't look anything like proper trimers. So we call them pseudo-trimers, or trimers in name only. And this is in clinical trials, amazingly enough. This is the basis of a vaccine that is in clinical trials, this one. But you can see it has not got a proper structure. So SOSIP trimers are used because they have a proper structure. And when you look at these, you see some interesting shapes. Here's one that looks like the little Tweety Pie bird. This one is the giraffe. This one is Bambi. And so we decided that we didn't want to have Bambi. We wanted to have a regular trimer that looks in the right shape. Here's essentially, you look at them and they look like a Jackson Pollock painting. So we've moved on from those old ones. Here's another interesting image. You can make it look like the you make it look like the Air Jordan. This is a trimer electron microscopy that looks like Michael Jordan. Here's one that looks like the Isle of Man, which is a little island off Britain. But we can have fun with electron microscopy. We can make some interesting shapes. But anyway, nowadays we can make these trimers from all of these different genotypes, all of these different clades, and when we make them to our design, they all look the same. So it doesn't matter what the genetic sequence is. We can make a clade Cs, we can make clade Ds, we can make clade Bs, and they all look the same because they all have a regular structure because we've stabilized them in the right conformation. And we can, here's on the background of the X-ray structure of the trimer, here's a ribbon diagram of one protomer, one of the 3 protomers. This is the disulfide bond we engineered between the subunits. Now, at the time we did that, we had absolutely no idea what the structure would be like this. But I'm not going to go back into the history, but we got it right. We worked out where to put it. And it turns out from the structure that it's in exactly the right place. This was done, the structure was done 13 years after we originally designed this disulfide bond, but we got it right. We can add additional stabilizing mutations here in V3, here in C1, and they further stabilize and antigenically improve the original version. So we can use the structure to make new design improvements that make a better version of the trimer. So it's a sort of cyclical process. Knowledge generates a new design. We can put in additional disulfide bonds. This is work in Rohir Sanders' lab. Peter Kwong at the VRC did a very similar design of an additional disulfide bond that gives even more stability. Here's another disulfide bond that can be put in. But at a certain point, you get to be stable enough. So, at what point are we good enough? If GP120 monomers are like this was our pretty good design. We can make an even better design, but are we going to finish up like this? Can we just over-stabilize these things? And so for routine purposes, this is the kind of design we use now, and we call it SOSIP V4. But we can go up to V8, etc., but they haven't generated a better immunogenicity profile. It's also important to purify these trimers correctly. So one of the steps that we developed in our lab, which helped us make these trimers, was use antibody affinity columns. So we can make monoclonal neutralizing antibodies and make an affinity column and then put a mixture of proteins down them and elute only the ones that bind the monoclonal antibodies. So we enrich for the species that we want. And if you use an antibody like PGT145, which only recognizes trimers, it is completely specific for trimers, you can take a transfection supernatant and in one column run, in 20 minutes, you can make a pure trimer preparation. So, and then you can do further purifications, you can run them down a size column, a size exclusion chromatography. But essentially, at least for research lab purposes, a PGT145 column will make a purified trimer preparation from a transfection supernatant in minutes. And when we make the trimers, again, this is work from Andrew Ward. You can generate electron microscopy images of where all of these different BNAbs bind. So these are all of these different colors are all different BNAbs that recognize different sites on the trimer. That one is the trimer apex, PGT145. As I say, it's completely specific for trimers. It will not bind to monomeric subunits. It will not bind to misfolded trimers. It is totally specific. There's the Manos patch, PGT121, 126, the CD4 binding site, VRC01, for example. The GP120, GP41 interface, which is another region of the trimer that only exists on the trimer. It will not bind to the isolated subunits. Antibodies like VRC34 and 8-ANK195, PGT145, PGT151, these recognize the interface. Now, we don't have the EMPA on SOSIP trimers. We truncate it here. So we lose the EMPA. We had to make some compromises. And in 2013, when we first made the BG505 trimer, Andrew Ward got a high-resolution 5.8 angstrom cryo-EM structure, and Ian Wilson's lab on the same preparation, or essentially the same preparation, made a X-ray crystallography structure. And the two of them were in very, very close agreement. So by two different independent methods, we got the same structure, and that was what was convincing people that we had the right trimer design. We don't have these problems. When you make other types of envelope, you finish up, when you think you've got a GP120 protein, and it should have this disulfide bond folding pattern, you get scrambling. A lot of the proteins in a GP120 preparation have scrambled disulfide bonds, particularly in the V1, V2 loop. This is work from Heather Deser's lab that was essentially completely ignored by the field for about 4 or 5 years. But she got it absolutely right, that you get scrambling, you get misfolded alternative structures. And some of the GP120s that went into human clinical trials of vaccines were these misfolded proteins. They were not even proper GP120 subunits. And you can finish up by making an intermolecular disulfide bond. So one GP120 will cross-link to another one. Or you can have a pseudo-trimer with different cross-linked disulfide bond patterns. Or you can get all sorts of stuff. You can get aggregates. That is not our problem. When we make a SOSIP trimer, Heather Deser assayed it for us, and there is no disulfide bond scrambling in SOSIP trimers. And that's why they assemble properly, because they're regular structures and they pack. So we're enriching for the regular proper structure. So we can make these things in vitro, but the major vaccine bottleneck is the cost and speed of manufacturing GMP proteins. So we may have a lot of proteins that we can make in the lab, but getting them through the FDA and EMA block to get them into human clinical trials is a major issue. So how do you scale something up? How do you make more of something bigger? I mean, if you're taking a drug, you just don't make a bigger tablet. You have to learn how to do this. So you have to have an in-house plan that will actually work in practice. And we worked with IAVI and in particular Antu Day to translate in Project Minerva. Minerva is a can of Spanish sardines. I don't know why, but we put 3 of them together to make our logo. And the goal was to make BG505 SOSIP 664 trimers as a GMP reagent. And we had combined expertise and support, and we published that paper last year in a biotechnology journal. And before we started it, many of our colleagues thought this is where we would end up, in a place called Shit Creek, which in English, which I don't know how it translates, but they didn't think we were going to do very well. They thought it would be really too hard for us to do this. But in fact, it turns out to be easier to make these SOSIP trimers than to make earlier generation monomeric GP120s or non-native GP140s. They're easier to work with. So we made a CHO cell line. We'd already made stable CHO cell lines in our laboratory, but this was made at a company. We transferred it to a second company who then made the protein. The protein was purified by GMP grade 2G12. We knew that it existed because it had been into human clinical trials as a GMP grade passive antibody. So I remembered that and remembered the paper from a colleague of mine, and we were able to buy the antibody from the freezer where it had been for 8 years and never going to be used, and we used it for affinity columns, followed by size exclusion, in a process adapted for GMP conditions. And we have to show that we can remove adventitious viruses to achieve a 10 to the 12 log virus reduction. And we use columns, membrane filtration, and detergent treatment. And all of the trimer properties in the GMP program were consistent with what we'd reported for the research grade material. There was no disulfide scrambling. The glycan composition and site-specific analyses from Max Crispin showed that the properties of the glycans were typical of what we expected, and we could do antigenicity assays and show that the BNAbs were present on the GMP product, BNAb epitopes. And this thing is highly stable. So this is what the trimers look like after incubation at room temperature for 3 months. and you take an electron microscopy image, and they're absolutely unchanged. They're like a rock. You can just leave them in a vial for 3 months, and nothing bad happens. And that's actually really very helpful. And we've done 5 degrees and 75 degrees, minus 75 storage conditions. We don't need special excipients. This is just in tris saline buffer. The only known vulnerabilities, if we put it in low pH, below 3.5, say, mix it with a Coca-Cola, it doesn't go very well. So acid destroys it, and hydrogen peroxide also destroys it, but that's not what you want to do to things. So we finished up with the trimers in a vial. Here is two members of our group holding the vial against a man with very hard abdomen. So if this is like a rock, that's like a rock, but we were not going to show our bellies because we do not have six-packs. So then we've got to say, well, what can we do with this? Well, we're running now with colleagues. Our colleagues are doing this. We're not clinicians. I am not a clinician. I cannot do this kind of thing. I would not be able to find a leg with a needle. But our colleagues are doing comparative immunogenicity trials now. How do the human antibody response compare to animal models? We can get B-cell information. We can make monoclonal antibodies. We're doing what I think is quite an important study to compare adjuvants. So which is the best adjuvant? We're working with Julie McElrath and the HVTN. We can get dosing data, and we can use it as a baseline, as a point of reference for germline targeting trimer, which is a much more interesting protein, a much more likely to be useful responses. So this baseline protein will serve as an integral control arm. And the first clinical trial, IAVI, Fred Hutchinson, and the Raygon, called the WOO-1 trial, started in December 2018. So what we've done with this trial is we've plowed the field. We've established that high quality trimers can be made in useful quantities, gram amounts, under stringent conditions. And the methodology can be adapted to the production of more interesting trimers, like the GT1.1 trimer. The VRC and the Shattuck EC consortium have now produced GMP trimers. The VRC trial started, I think, 2 weeks ago. That's a very similar, it's BG505DS SOSIP, which is very similar to ours, but they're doing a different adjuvant. And all groups have made these trimers in CHO cell lines, essentially. So again, this was a collaboration between the IAVI, and Antu Day was the key player. Al Kupo from my lab is the person who understands translation. Pervin Ankelsaria from the BMGF, and Jim Braddock, who's always supported us at the NIH. And so all of these groups and organizations worked together, drove this to a successful conclusion. Now, we have a really difficult task ahead, though, because HIV envelope proteins are very poorly immunogenic. Here are binding antibody responses to SOSIP trimers in red compared to influenza HA trimers in Kymab mice. This was a mouse model. And you see how poorly immunogenic as a binding antibody titer HIV trimers are compared to influenza trimers. That was done with soluble proteins. If you do mRNA prime, protein boost, there's the response to HA. Here's the response to HIV envelope. So we have a terrible immunogen. And this is not just SOSIP trimers. This is all HIV envelope proteins. Influenza is really easy, and this in comparison. And this is why we need adjuvants, because we need strong adjuvants. And cryo-electron microscopy shows how the envelope glycoprotein is shrouded in glycans. These are all the glycans on the outside. And I'll show you the antibody epitopes are holes in the glycan shield. But all of this outside is glycans. So there's the basic structure from Andrew Ward, and the glycans go on top in green. And you can see how little gaps, how few gaps, and how small they are between the glycan shields. So the glycans cover the protein surface, and there are a lot of glycans. There are 32 glycans on a GP120. There can be 90 glycans on a trimer. It shows you just how difficult it is to get past the glycans. And now the Max Crispin's lab and Jim Paulson's lab have really taken these SOSIP trimers and measured every glycan in great detail. How occupied are all of these sites? Are they high mannose? Are they oligomannose? What do they look like? Because the glycans contribute to antibodies. So glycans are immunologically self. It's a wolf in sheep's clothing. This shields. This is wool. It shields the wolf underneath. And the glycans are not recognized. They're generally very poorly immunogenic. But the immunogens must contain these glycans because the glycans affect the protein epitopes, and some of the glycans contribute to the protein epitopes. So some of the glycans are components of neutralizing antibody epitopes. So we have to have the glycans. But they block access to the epitopes, but they also contribute to the epitopes. And some BNAbs can recognize and penetrate the glycan shield. So here's the N332 glycan, which is a major site of BNAb binding. There are a lot of BNAbs that will bind to protein surface plus this glycan structure. So you need the right glycan structure in the right orientation and of the right composition. And N32 binds to a region of V3, and the N301 glycan is involved. This is the PGT128 Fab, but there are many other Fabs like this. So you cannot ignore the glycans. You have to have the glycans in the right place, in the right orientation, and the right structure, and this is another major part of the problem we face. So this is the Ascent of Mannose, not the Ascent of Man, the Ascent of Mannose. Mannose content is important on trimers. If you look at GP120 monomers, the early generation proteins, 36% of the glycans are in the high mannose form. A non-native GP140, 35% of the glycans are in non-native, 35% of the glycans are mannoses. But you get up to SOSIP trimers, 68% of the glycans are in high oligomannose. And the GMP grade trimers have this 68% mannose. So the mannoses are not very well processed, but they're different than the glycans that dominate on the GP120 proteins. And you have to understand how these are made. The glycosylation is affected by trimerization. And the mannose patch, this is what we call the integral mannose patch that is present on GP120, but the TAMP is the trimer-associated mannose patch, which is unique to trimers. So the high oligomannose TAMP region is only present on trimers. So glycan clustering stabilizes the intrinsic mannose patch, the IMP, which is this region. Native-like folding generates the trimer-associated patch, the TAMP, and glycan clustering preserves the vulnerability to broadly neutralizing antibodies. So this is basic science knowledge that we can derive from studying these proteins that is really important for immunogen design and for defining how BNAbs evolve. And it comes down because processing enzymes, mannose patch is created because of a processing enzyme called monosidase. And if monosidase enzyme cannot, that's the monosidase enzyme in blue, and if it cannot get at its substrates, it bounces off. It's a steric clash. And on the trimer, the monosidase enzyme cannot properly process some of these glycans, so they stay in high mannose form. That's the mannose patch, that's monosidase attempting to digest these glycans, but it fails to do so because there's a clash. It just cannot bind properly. So you finish up with high oligomannose structures that contribute to broadly neutralizing antibody epitopes, but that shield it. And again, this is basic knowledge. This is from Max Crispin's lab, done using our trimers as a collaboration. It teaches people a lot about how antibodies recognize proteins. But an antibody like PGT128 can recognize this trimer-associated mannose patch. The antibody has learned to accommodate and fit between N301 and N332 and penetrate to the protein surface. So the antibody epitope is formed by a bit of that glycan, a bit of that glycan, and some protein surface. And it will not bind without the glycans. So again, if you want to induce this type of antibody, you need to present the glycans in a way that is present on a trimer. Glycan underoccupancy. So it's not every glycan site is fully occupied. So you can find, this is data from Max Crispin, but it's broadly similar on Jim Paulson's paper. So some glycans on the SOSIP trimers, particularly in V1 and V2, are not fully occupied. So the site is there, it's a NXT or NXS site, but the sugar is not fully added on all of the trimers. It's more occupied on the virus than it is on the SOSIP trimer, but it's still not bad. I mean, there's pretty good resemblance between the SOSIP trimer and virus-on. This is virus-on from Jeff Lifson's group. So the SOSIP trimers are not perfect representations of what's on the virus because some sites are not fully occupied, but they tend to be in not very interesting regions of V1, so we're kind of not too worried about that. So under-occupancy of SOSIP trimers has not affected any key BNAb epitopes. So we've seen under-occupancy, but not in BNAb epitopes. But it may create immunogenic glycan holes, and we can improve this. We can improve glycosylation. Rohir Sanders and Max Crispin working together have found ways to overcome the problem. We need to be careful when we're making large amounts of these in high production systems because we could run into a problem, so we've got to watch out for it. So, but how do you develop a good immunogen? How do you know what is a good immunogen? We can make good antigens. Proteins are antigens, but we want the immune system to respond to these proteins. Well, how can we tell what's a good immunogen? Well, we can't. The rabbits tell us we have to let the animals speak. And if the animal says this is a good immunogen, we listen to that message, but we need the animal immunogenicity data. And here's an experiment that was from our first paper on BG505 trimers. This is the autologous antibody response to the ability to neutralize the same resistant virus, tier 2 virus. A tier 2 virus is resistant to neutralizing antibodies. And anyone can make antibodies that neutralize the easy lab-adapted viruses. I mean, that's trivial. We could do that 25 years ago. But that's not a solution. We need to neutralize the hard-to-resist tier 2 viruses that are transmitted between people. So this is rabbits immunized with BG505 SOSIP trimers, and we get a strong, it's inconsistent, but it's still pretty good, neutralizing antibody response to BG505. That is a neutralizing antibody response to the B41 trimer, which is a clade B trimer. So it's true of two different trimers. But if we make a non-native version of BG505, or look at other proteins that have been published in the literature, they all fail to generate the neutralizing antibody against the tier 2 viruses. So the proteins with this bad shapes that I told you about, they fail. The proteins with the good shapes, they succeed in generating the tier 2 viruses. And we can map these epitopes by making virus mutants and seeing which virus mutants are resistant to the antibodies we induce. And then we can plot those epitopes on the structure of the trimer. And Andy Ward can generate electron microscopy images of the antibodies bound to the trimer. And there's one trimer we call the 241289 site. That is a hole in the glycan shield that is immunogenic, and antibodies are induced to it. There's another site we call C3465. Another group now calls it C3V4, but they've made monoclonals to this site. A few animals respond to V1, but this is all mapped data. So these are Fabs bind to the C3465 site or the 241289 site, antibodies to the interface fusion, and these are non-neutralizing antibodies to the trimer base. We don't want these. We're trying to get rid of these. These are highly immunogenic, irrelevant antibodies because the base of the trimer is immunogenic. So this was all published, mostly published last year. So we can induce autologous antibodies to the same virus as the vaccine strain, but in vivo, you can induce autologous antibodies that then undergo escape and somatic hypermutation and sometimes leads to BNAbs, broadly neutralizing antibodies. So we can do this, but we have not done this. And no one has done this. No one has taken a vaccine and taken it from here to here. This is the really difficult thing that we're having to try to do, because no single protein like these trimers is going to do this by itself. It's not going to happen. We need more sophisticated regimens. So we have strategies including germline BNAb targeting, particle presentation, lineage of vaccines. Not every group can do every approach. So a group our size can only do some of these approaches. So we're making germline targeting trimers, and we have what we call the GT 1.1 trimer that is now in a GMP production program. Others are being evaluated in knock-in mouse models. We want to present trimers on nanoparticles, and we're testing new protein-only or non-protein designs. We need to block the non-neutralizing epitopes at the trimer base, perhaps. We're working on that. We incorporate immune modulators into the design of soluble trimers. We have to identify the best adjuvants in preclinical and clinical design. So all of these trimers here look alike. I showed you this kind of thing earlier. These are all from different clades, A, B, C, D, E, H, X4 virus, R5 virus. They all look the same. But the immune system does not respond in the same way. So what predicts trimer immunogenicity? Again, the rabbit or the macaque tells us what the answer is, not the electron microscopy. So a native-like trimer, maybe these native-like trimers, I showed you, they're necessary, but here's one native-like trimer, doesn't induce an autologous response. Here's another one, induces a weak one. Here's the BG505 trimer, induces a very strong one. But at the structural level, these three trimers in the three different colors, green, blue, and black, they're virtually identical. So it's all the subtle presentation of epitopes that makes the difference between an immunogenic and a non-immunogenic trimer. That is the same thing on a larger scale. This is multiple different SOSIP trimers. Some induce very strong and consistent autologous responses, and some are as immunogenic as a brick, and we don't want bricks. And there's just nothing in vitro that we can measure that will determine what that outcome is going to be. We sometimes do see neutralization of tier 2 viruses. It's not particularly strong, but it's not zero. But we don't have a recipe. So here's some trimers. These are BG505s. These are highly stabilized from Rohir's lab. Sometimes we get red hits in our neutralization assays done by David Montefiore. But we need to get this all red across the board and have a recipe. We need to know exactly how to do this. So we can do it sometimes, and sometimes isn't yet good enough. We have an adjuvant testing program. I said we're working with Julie McElrath and companies. These are eight different adjuvants in rabbits. These are the autologous antibody titers. This is guinea pigs, sorry. These are the autologous antibody titers, and these are cross-neutralizing tier 2 titers. So some adjuvants are better than others, and that might be important. But these are still quite low. These are titers around below 100, whereas these are titers above 1,000. But we're doing a preclinical studies, and we're going to do human clinical studies to try to see which are the best adjuvants. Germline targeting trimers. So this is Rohir's lab. They spearheaded it in Amsterdam. And we took the BG505 trimer, which doesn't recognize the germline precursors of BNAbs, the proteins that need to be recognized in the germline of the human genome. And Adrian McDermott talked about this yesterday with Lionel Messi and some fat guy kicking a ball around. So this is the fat guy kicking a ball around, and we need to make the Lionel Messi type of germline BNAb binding. So by rational engineering, amino acid changes were put in to make the GT1 trimer. And here you've restored in green the epitope for some of the BNAbs to the trimer apex and the CD4 binding site. And then you can take that further and make some mutations and make even stronger binding to these germline BNAbs. And this is the GT1.1 protein that is in GMP. And you can make a GT1.2 that has other properties. So by rational engineering and amino acid changes, you can create the ability to bind a germline BNAb. And these are now in evaluation in knock-in mouse models with Fred Alt, Facundo Batista, Laurent Vercosi, Michel Nussensweig, and we have a macaque experiment in wild-type macaques with Francois Villinger and Bali Palendaran. And we need to induce these BNAbs and then polish them and mature them using various trimers. And this is an experiment that Rohir did with Laurent Vercosi. So you do in a VRC01 light chain animal, knock-in mouse made by Fred Alt. And you see, after one immunization, you see some induction, but then you can put in a second trimer and get better induction, and a third trimer and get better induction. And in the end, you can get better induction of BNAbs in these knock-in mouse models. But you can't, in practice, have a human vaccine regimen with 5 different trimers. So we've got to understand this and simplify it and figure out what to make an optimal practical design. We also need to understand antigen presentation pathways for soluble trimers and for nanoparticles. We're trying to make nanoparticles. Nanoparticles may be better immunogens. It's not really yet proven. But this was taken as a Darrell Irvin paper in Science about 2 months ago and as a commentary. And it shows that the nanoparticles, are presented to the immune system differently than soluble trimers. And that's interesting. So we have to figure that out. So we're all making nanoparticles of different designs. Darrell's design is different than the one we're working on. And here's another design that we're working with Neil King and David Baker. This is a protein-only design, and this is cryo-electron microscopy of the I5350 particle. These are SOSIP trimers on a protein nanoparticle core. And you can get a nice electron microscopy image on them. And we can do immunogenicity studies with these and understand them better. And here's an experiment with nanoparticles displaying SOSIP trimers. This is a nanoparticle design we've made in our lab. It's made by chemically coupling SOSIP trimers to a solid phase, a solid sphere of 25 nanometer diameter. So it's chemical coupling. And the trimers on the particle bind the PGT145 epitope OK, they bind the VRC01 epitope OK, but they don't bind this irrelevant epitope. So the trimer particles are the same antigenicity as the soluble trimers, so we can couple them properly. We can couple the GT1.1 trimer onto nanoparticles. These are 4 different germline BNAbs binding to the nanoparticles. So we can make nanoparticles. Now we have to learn how to use them. We can put 2 trimers on the same particle. We can put a red trimer and a blue trimer on the same particle and detect them by a red-specific antibody and a blue-specific antibody. And you only get a signal in the ELISA in these circumstances where you capture with one and detect with the other. Otherwise, you don't get a signal. So we can couple 2 trimers to the same particle. We can probably do 3. We might be able to do 4. And trimers, bivalent particles, might be useful. This is a mouse experiment that's still ongoing. This is mouse immunized twice. We do 2 doses of B41 particles, and there's soluble trimers. There's trimers on nanoparticles. It's about 200-fold better binding antibody response. There's soluble trimers with a T helper epitope tag. That is a better response. Particles on a nanoparticle, trimers on a nanoparticle, and with the T helper tag, and you get a really strong binding antibody response after 2 immunizations. But is that going to translate to a better neutralizing antibody response? Well, we're still working on all of this. This is still ongoing work. But, in terms of basic immunogenicity, particles are generally boosters, but we have to figure out how to use them. And the nanoparticles, this again is the antigenicity. We've blocked off the trimer base. This is an antibody to the trimer base. So it binds to the soluble trimer, but it doesn't bind to the nanoparticle. So we can block off this immunogenic, irrelevant epitopes that we don't want, but we retain the VRC BNAb epitopes that we'd like. And in the mice, the immunogenicity of the trimer base is reduced in the nanoparticles. So that's antigenicity in vitro. That's antibody response. we get inhibition of anti-base antibodies. That's the anti-trimer antibody to the soluble trimer to the particle trimer, so it's greater in the particle trimer, but the anti-base antibody is less on the nanoparticles, so we can block off the base. So I'm finally just going to return to thanking people. This is thanking Ian Wilson for and Andrew Ward for the amazing structural biology they do. I've worked with Rohir Sanders since he was a grad student with me in 1998. I gave him, this was him as his PhD ceremony. Over the years, he's lost a lot more hair than I've had. So I've kept my hair. He's losing it really quickly, and he's 20 years younger than me. So, you know, bad luck. And so we also have friends that we work with. This was all taken at, Rohir was promoted to professor in Amsterdam 18 months ago. So we all flew across for his promotion ceremony. And when they do a Dutch promotion ceremony, they all dress up in these clothes, and we have to walk through the town and go to a ceremony, and it's all in Dutch. But hey, it was fun. And then we had an awful lot to drink to celebrate his promotion. So I'll leave it here, and I'll take any questions. Thank you.
Thank you, Professor. It's a very interesting lecture. And we are open for questions.
So, John, I know this is a naive question, but I'm going to ask it anyway. So, ultimately, you're trying to create something that's like a virus particle in terms of its immunogenicity. So, if you do side-by-side experiments in terms of measuring antigenicity and immunogenicity, are the SOSIP trimers, whether they're linked through disulfide bonds or whether they're on a particle, are you getting to that same level of antigenicity and immunogenicity that you can get with just a virus particle?
Well, in infection, in around 20% of people, BNAbs emerge. And it's complicated. I mean, it takes multiple years. You know, nobody develops a BNAb in 3 or 4 days. You know, BNAbs emerge in 2 to 4 years or longer. Multiple cycles of virus escape, multiple cycles of somatic hypermutation, multiple consistent presentation of immunogen. You make a single protein of a single genotype and stick it in 3 times, you're not mimicking that process. So that's why we're not going to induce BNAbs just by making a trimer and stick it in your arm in an adjuvant. It won't happen. We have to better mimic the natural process, which requires somatic hypermutation and germline triggering. And making particles might help with that or might not. And you develop BNAbs quicker in infected infants. So we're doing some experiments with Sally Permar in an infant macaque model to see if we get a better response with germline targeting in infant macaques. So, you know, no, we can't do as well as you can generate in natural infection in a minority of people. We can't achieve that. But we have to figure out how to achieve that.
John, with the PADRA tag, working with Pierre and Michel, there was some improvement in immunogenicity, and focusing on thinking about the T helper cells that you might want to induce at the same time you inject the purified SOSIPs that you have. Have you or has anyone looked at those sulfide bond failure products to see if they will induce CD4 T cell responses?
You know, and it's entirely possible. And T helper responses to envelope are terrible because the T helper epitopes are shielded by glycans. They're either directly shielded by glycans or it screws up the processing enzymes that liberate the T helper peptides. And this has been incredibly poorly studied. I mean, the number of papers that have been done on HIV envelope, very few of them are focused on the immunology of it. And the most detailed papers were actually done about 15 years ago, which is pretty ridiculous. So I'm conscious that there's a lousy T helper epitope problem. So we've grafted PADRE or DPT or TT tags onto the C-terminus of trimers, and we haven't got the animal resources to test all of these. And whether an initial binding antibody response will truly be beneficial, I don't know. But And of course, adjuvants induce T help, which is another complication for interpretation. But I think we need a better T helper response. I absolutely think it's one of the reasons why it's a lousy immunogen. So yeah, you suggest putting in as well a GP120 monomer that will be less folded and more easily processed, and perhaps that would. But T helper responses have not been induced well by GP120 monomers in the past. So the Padre tag has cathepsin cleavage sites either side. We don't know if we're liberating it, but you see something in these mice. But is it something that's meaningful or just something?
Mais alguém? Okay, so thank you, Professor,
Thank you.
very much.